All right. So it's my pleasure to introduce Dr. Marcela Uliano-Silva, who is currently a senior bioinformatician at the Wellcome Trust Sanger Institute, or Wellcome Sanger Institute, it's called now. And she's working on assembling genomes of eukaryotic diversity of Britain and Ireland in the Darwin Tree of Life project. So she holds a PhD in the biological sciences from the Federal University of Rio de Janeiro. And she previously, like prior to the current role, she was working in the Leibniz Institute for Zoo and Wildlife Research in the Berlin Center for Genomics and Biodiversity. And I know her because she's been part of the Vertebrate Genomes Project for a long time, and where she in particular contributed the assembly of the two-toed sloth. So yeah, she's quite active in the world of genome assembly. I'd say that Marcella is an explorer of new and difficult problems that have come up in the process of trying to understand the scope of the whole tree of life. And she's also been a guide and has done a fantastic job at communicating her work and expanding the scope of genome assembly, for instance, in working on improving the mitochondrial genome assemblies in these kinds of projects. She actually has some bioinformatics software that is designed specifically for that. And yeah, so it's great. We really wanted to have her interact with the department because we have a lot of problems of how to understand and curate assemblies that we're making. We're increasingly making long, complete assemblies of genomes and using them in our research. And so we really look forward to the perspective that you can offer, Marcella. And with that, please take it away.
Eric, thank you so much for the very kind words. It's a pleasure for me to be here. So thank you a lot for the invite. Right, so I will present to you a little bit of our project, the Darwin Tree of Life project, and I will talk about some of the many scientific outcomes that came out of it as well, and the ones that I'm more involved in. So yeah, so Eric just introduced me, but I will just go through this very quickly. So yeah, I'm from Brazil. I did my bachelor, master's, and PhD there, then spent some time in Berlin, and then I moved to the Wellcome Sanger Institute as a senior bioinformatician. I have worked in the assembly team for 4 years, and now I have transitioned to Mark Blackster's team, but I'm like highly involved with assembly, and we collaborate and we are all together. It's just because there is two sides of the science that I do. One is assembly and software development for genome assembly, and the other one is comparative genomics. And I've been focusing more on this now. So then I'm on Mark Blackster's team at the moment. But we are a very big group and we interact a lot. So there is a little video about the Darwin Tree of Life project. We have time, so I think I will show you, because videos, it's nice for you to see the environment, to see how the Sanger Institute is and how the Tree of Life program is running. So I will just play. It's 3 minutes. Ooh, okay, so I think I need to go out of the presentation mode and do this. Let's see.
Blackster. I'm head of the Tree of Life program at the Wellcome Sanger Institute. Our goal is to sequence the genomes of all the fungi, plants, animals, and protists on this planet, both to understand how they've evolved, how they work, but also to use that information to conserve biodiversity and to promote human health. The way we're approaching this huge project of sequencing everything is by starting on a number of named projects. So one of these projects is the Darwin Tree of Life project. That project is to sequence everything that lives in, on, and around the islands of Britain and Ireland. This is a collaboration with people across the UK in museums and botanic gardens and other institutes to collect specimens for us. So the botanists at botanic gardens know where all the species of plants are. The marine biologists know where to go on the beach and where to dive in the cold waters of Britain and Ireland. And the Naturalist Museum and their army of collectors know where to go to find all the species that we need to sequence.
I'm Ines Janishczak, and I'm the sampling coordinator for the Darwin Tree of Life project at the Natural History Museum. We're currently in Millport, and the idea is we want to sample a wide range of locations around Millport for marine species. Our main focus is invertebrates. So we're basically trying to get a snapshot of what's here so we can add it to the project and increase our species list. In the lab at the moment, we're actually just going through every single individual tray and we're picking out all the things that look interesting. We have an ID team who are going to ID it to the best of their ability. We then process them at the Natural History Museum and then they are sent to Sanger for whole genome sequencing if things go well.
I'm Graham Oatley. I'm an advanced research assistant in the Tree of Life core lab. We work in a laboratory and taking the samples that we get from partners, extract DNA and RNA from them, and then pass them over to the sequencing team. At the moment, there's no one-size-fits-all extraction protocol. So the key challenge is to try and develop different extraction protocols for the different organisms that we're getting in to get the high-quality genomes that we're aiming to sequence for the Darwin Tree of Life.
I am Marcel Uliano-Silva, and I am a senior bioinformatician in the Darwin Tree of Life project. So after the samples been sequenced, it comes to our team, and we assemble the genomes of all of the species that come through. So assembly is basically the process of taking the shorter DNA sequences and finding overlaps between them with the help of computer software to build up ideally chromosomes. So genome assembly is basically like assembling a jigsaw puzzle, which varies in shapes and sizes.
I'm Charlotte. I'm a PhD student in the Darwin Tree of Life project, and I'm studying the evolution of butterflies and moths. Moths and butterflies are extraordinarily diverse. In fact, they are 10% of all described species. And in the past, studies that have looked at genomic data have been limited to perhaps 3 species. So you can say what's true in those. But as part of the Darwin Tree of Life, we've been able to sequence hundreds of these Lepidopteran species. And the interesting thing that we've found so far is that Lepidopterans are kind of a story of 2 sides. On the one hand, most species have undergone very little change, but in a few cases, they've undergone dramatic changes. They've stuck together bits of their chromosomes and then split them up again, which means their overall genome looks very different. And we're really interested in what has allowed them to avoid this constraint that the rest of Lepidopterans face.
My job here at the Tree of Life is my dream job. Ever since I've been a scientist, I've wanted to understand organisms at the genomic level. And every time I looked at a new organism, I wanted to look at more. And so suddenly here I am with some amazing colleagues able to do that, to sequence at scale across biodiversity. And that to me opens huge vistas. Rather than understanding how this one species does what it does, we're going to understand how all species do what they do. And I believe that this is going to change the way we do biology forever.
Right, so I think it's always nice to have a look at this video and shows the Sanger and shows our project a little bit. Am I passing the slides? Do you see the Earth Biogenome Project now? Okay, good.
Yeah.
Right. So the Darwin Tree of Life project, as the video mentions, is one project among many projects that are happening to sequence lots of biodiversity on Earth, right? And all of these projects are under the umbrella of the Earth Biogenome Project, which wants to sequence all eukaryotes, extant eukaryotes in the planet. So we have now consortia such as the Darwin Tree of Life project, which are geographically based. So the Darwin Tree of Life project is Britain and Ireland. But we have some other types of projects. I think Eric mentioned the Vertebrate Genomes Project, which wants to sequence all the vertebrates. We have the European Reference Genome Atlas, which is also region-based. And then on the top of all of these projects is the Earth Biogenome Project. So I guess for a lot of biologists, I don't need to necessarily justify why we are sequencing everything, right? So the sequencing costs are very low. We have good technology now to produce reference genomes, and the knowledge that we have of the biology on the chromosome and genomes that we have at the moment is very little, actually, that we be able to grasp in terms of the human genome and other modern organisms that we have sequenced. So basically, to really answer fundamental questions in evolution, to understand how chromosomes evolve, to understand what are the conserved blocks across the Tree of Life, and what are the very the blocks across the tree of life, they are broken and they're taxa specific. You do need to sequence everything for us to unravel new biology. And then these genomes are also going to help us with clade-specific adaptative questions. And at the same time that we do all of this evolutionary biology, which is my main interest, I think is the biggest point of the project, we are also yielding good reference genomes for conservation projects that need those genomes. And in the process of sequencing everything and producing all of these genomes, we have been using several methods to advance the field of genomics and bioinformatics as we move the project along. And one nice thing about sequencing Britain and Ireland is that these islands hold lots of different species, even though it doesn't seem like it. And they represent around 40% of all described families of species. And this is a very good, deep phylogenetic signal there for us to actually really study evolution of these lots of different clades across the tree of life. So as the video showed, we have many different partners. So it's not only the Wellcome Sanger Institute. The Wellcome Sanger Institute is doing the extraction of the DNA, library preparation, sequencing, assembly, and curation of these genomes. But we are taxonomists, field scientists, wet lab scientists, evolutionary biologists, in lots of different institutions, which really creates this chain of biological science work, which is not only bioinformatics, it's field scientists related. They are all empowered and employed within the project to make this project happen, which I think is really important. So our goal is to generate high quality chromosome complete reference genomes for all named eukaryotic species that are native to Britain and Ireland, to annotate these genomes and to release them openly by the use of all. So the annotation of these genomes is done by the EBI. So as the genome is sequenced, assembled, and curated by the Sanger, it's released openly to the public INSDC databases, and then the EBI picks them up and annotates them and also releases the annotations publicly. I will show you very quickly our tree of life genome sequencing engine and assembly. So it starts from the sample acquisition, right? So we have the field scientists, they collect, they have tubes with barcodes, and they record all of the metadata necessary before and after the collection. And all of this is registered in COPO, which is a metadata database. Then there is the morphological identification by the experts of this all the species that come through. And then we do also DNA barcoding validation. So we sequence more than one data type for all of the species that come. So it's PacBio and Hi-C mainly, and then we have RNA-seq for annotation as well. So imagine we have thousands of samples coming. We need to make sure that we don't have sample swaps, between the tubes and also between the different data types that we produce. So we have bioinformatic pipeline for DNA barcode validation. Then very quickly, then after that, we do the sequencing, library prep and sequencing, and then it comes to the assembly team, and we do assembly and scaffolding, and we'll talk a little bit about Hi-C as well. After genome is assembled automatically by the assembly team, then it goes to the curation team, because there is lots of tiny things that automatic pipelines cannot do still to assemble chromosome level genomes, and then the curation team does manual curation to raise the standard of these genomes. Then this data is all submitted to INSDC, and then the EBI EMBO is annotating these genomes. They are open release on INSDC, so NCBI and portals, and we also have a Darwin Tree of Life portal. And all of the species also have a genome node publication. So people using these genomes can cite this publication and look at all the metrics that were involved and how these genomes were produced. So here is just to show a little bit of the nice work done by the field scientists, collecting species in remote places. If you want to know more about the work of our collaborators in this project, you can always access the blog from the Darwin Tree of Life and see what's going on lately. So PacBio Hi-Fi data is at the core of the DTOL. So we have lots of different PacBio sequencers, including Revios, which gives very big yield of very like long enough reads with very high quality per base, which help us to assemble very high quality genomes. But we also have, as I said, RNA-seq done with Illumina. Sometimes we sequence Illumina short reads as well. And we have the chromatin conformation structure, the Hi-C data that is used to do scaffolding across centromeres, if the species have centromeres. So across, like very long repetitive regions, and then to construct chromosomes. And we do some tests with nanopore as well, but the center core is PacBio for the Darwin Tree of Life project. And as you can imagine, in the lab, there is a lot of development happening because we are sequencing across the tree of life. So it's fungi, plants, lots of the different species. They are non-model organisms that don't have standard DNA extraction and sequencing protocols. So the Tree of Life core lab has each species needs one scientist at least to be there and to extract a DNA. This is something you cannot automate like from the beginning to the end. So they work really hard. And I just want to point you that they have released lots of the protocols they use for very difficult samples. I think like lots of people can benefit from this because there is a lot of people around the world trying to extract high molecular weight DNA from different species, and they have released openly, publicly their protocols, and they are also very easy to reach out. They are also welcoming chat and people reaching out to them to talk. So as I told you at the beginning, I've been part of the assembly team for 4 years. Now I transitioned, but these are some of the people on the assembly team. And I will talk about this very quickly because I know you are interested in this. So just show you very quickly our assembly pipeline, okay? So it starts with PacBio Hi-Fi data. So PacBio Hi-Fi data is long and has high quality per base, basically because you can insert this adapters in in the double strand sequence, and you can have many passes on the sequencer. And then basically you make a consensus out of the sequence, which gives you a high quality per base in long reads, which will be 6,000 to 15,000 base pairs long. And these reads are then inputted to assemblers, right? So we use HiCanoe and HiFiASM being the one that is most used. Then because these reads are very long and very high quality per base, if you have a diploid genome, which like 50% of the genome comes from the mom and 50% from the father, then you are able to separate these haplotypes, what we call, so both haplotypes from both parents very well, because the reads, you have variation enough, the SNPs enough that helps you to disentangle these two haplotypes. And then we use a tool called purge dupes to separate these both haplotypes. And then after we have these two versions separated, we take one, which we call primary assembly. So one reference only for one of the haplotypes if it's a diploid species. And then we do the chromatin conformation structure scaffolding pipeline, which basically it's a technique that can freeze the information of the 3D structure of the chromatin in a somatic cell. So a chromosome that is like, when it's linear, it's very long. When it's like in the somatic cell, we can have contacts with itself, and then we can freeze this information sequence as short reads, and then we can basically use the mapping of these reads to scaffold across the chromosome and build up chromosomes. And then we use this pipeline, this tool called MitoHiFi, which I developed to also assemble the mitochondrial genomes of the species that we sequence. And this pipeline, basically PacBio Hi-Fi and Hi-C, has been working very well across the Tree of Life, and I will show you some of the results that we have. So this is what we call out-of-the-box genomes. Okay, so those are Lepidopteran genomes, moths and butterflies. So if you have never seen a Hi-C heat map, I will explain you very quickly. So these heat maps here, you see these red squares in the diagonal. These would be chromosomes of an assembly of a genome. Okay, so for the species that is on the left. So basically, if you see a very intense red in the square and nothing off of the diagonal, it means that all of the Hi-C reads are mapped back to that scaffold, which in this case is a chromosome, and we don't have any misassemblies or errors. So basically, we have all of the chromosomes assembled really well, and the high C shows us that. But this is not what happens for a lot of other species that we have. So I'll just give you an example here. And this is why we have a curation team. It's still essential to do manual curation. And this is a case where Joe's Wood's team, which is called the GRID team, which is the curation team, one curator needed to do more than 1,000 joins manually. And you have a high C plot before on the left, and then a high C plot after they did the manual curation on the right. You can see the chromosomes resolved. There's lots of other signals you can see in high C as well. So this like orange, they are shared between different scaffolds, are like biology showing up on the high C plot. It can be repeats, they are shared between the chromosomes and so on. So there's a lot of things that you can see on high C maps as well. So here is also just showing you, we go from this to this many times with manual curations because the automatic pipelines cannot do the scaffolding by themselves. So with this pipeline and with our engine, we can do the very large. So we have sequenced the mistletoe genome, which is 94 gigabases. It has broken all of our automatic pipelines, but we got it through. But it is challenging. So tools like some tools and other things cannot index very large chromosomes and so on. So it is a challenge for the bioinformatics tools that we have available. We can also do the very little. So there is a protocol called PIMS, which was developed by Chris Lamar. He's now at the Natural History Museum, which can get like DNA from very, very tiny samples. And they have been using this in lots of different species. So one example is the work that Erna King does with some very small nematodes, which have like 1,000 cells per animal. And they are being able to extract DNA from one individual for PacBio, and then they do pooling to do the high C to do the scaffolding at the end. So we do the very large, the very small. We also assemble more than one genome per sample. So again, because of the quality of the PacBio reads, we are able to extract cobions also out of the genomes. So you have a moth genome and you have the Wolbachia complete genomes also sequenced and assembled out of it in the mitochondria, which is also really nice. So I want to, just show you that we have lots of different portals and different tools available and, data being produced, and you can have a look. So you can have a look at the raw data and assembly progress in our TalkQC pipeline, the ensemble annotation browser with the species that are already annotated. You can find it all there. And we have the portal for the project. So I can give you the slides and you can go through these different portals later that the project is producing. So we have published as genome notes in the Wellcome Open Research 550 papers in the collection so far. We have more species sequence, but the genome notes are, they come a little bit after. So you can all find all of these species there. At the moment, so this is the numbers we have so far. As of 1st of February, we have produced 1,395 species that are complete and public. And we are working on 3,500 species across the tree of life. So we had an initial bias to Lepidopterans especially because of COVID. So there was a limit on what we could collect and where we could go to collect. So we were able to collect lots of Lepidopterans, and also because they go through the pipeline really fast. But now we are getting on to like more challenging taxa as well. Right, so I present you the project, and now I want to show you a little bit of some more of the science. I already showed you some of the science that is coming out of it, but I will show you a little bit more of some science and development that is coming out. I think one Eric has mentioned is MitoHiFi. So this came basically, we had all of this PacBio data, and I was assembling it and looking at it, and all the time I was finding mitochondria contigs. It was like, oh, it's the mitochondria. And then you see that, so mitochondria, if it's a circular mitochondria, de novo assemblers might have this they because it's a circular molecule, they end up doing the overlap or the debridging grafts and just assembling the mitochondria redundantly many times. So you can have like a mitochondria is 15 kb, but then you have a contig that is 30 kb, because basically the overlaps just found the other part of the mitochondria and just assemble it again. So many times, if you find a mitochondria contig, you need to basically remove the redundancy. So I was there assembling nuclear genomes, and then I was finding this mitochondria contigs. I was like, okay, so now I will write a script to kind of like remove the redundancy. And then from that, you start building up, and then we end up with a pipeline that basically starts from the beginning to the end, assembling the mitochondria, right? From if you can give PacBio reads and you need a reference, and then you will assemble all of your mitochondria. So MitoHiFi is able to assemble lots of different mitochondria across the tree of life, including very large fungi ones. But it is challenging mitochondria and chloroplasts. Are not the best with MitoHiFi, which needs a reference, so it's reference based. So ChainG has been working on a tool called OTK to assemble large plant organelles. So mitochondria of plants have many repeats, and the chloroplast also has many repeats. The tool is actually available. The paper will be coming out soon, so this is reference free. It works really well. Right, so this is something else. So it's not Darwin Tree of Life, but it's a partnership with Tree of Life as well. So this is João. I just want to show you his work. He's my PhD student. I'm his co-supervisor, and he works with biological invasion. So we did the golden muscle genome. So I did that also as my PhD project, but it was an Illumina assembly at the time. Now we did the genome with PacBio Hi-Fi. And he's also working with some polyploid corals. So the golden mussel and the corals are invasive in Brazil. They mainly use the good quality reference genome to do population genomic analysis of the different populations dispersed around different areas of Brazil. But also the golden mussel, they would like to be able to develop a biotechnological strategy of control. So the problem is similar to the zebra mussel, I guess you are familiar with. And now we have high quality genomes for them, and João is working really hard on his corals as well. This is another work I've been involved in, and is also in the context of the Vertebrate Genomes Project, which are Sicilian genomes that we have sequenced. And it's really nice to be in this moment that we are able to sequence chromosome level genomes for taxa that have very large genomes, as amphibians do, and the Sicilians do have also very large genomes as well. As it turns out, more than 69% of their genomes are made out of repeats. But one thing that was very interesting, so Sicilians are limbless amphibians, no? And as soon as we sequenced their genomes and we had them complete, we were able to look for the specific enhancer of the sonic hedgehog, which is the ZRS enhancer, which is missing in the Sicilians. And this enhancer is also missing or mutated, highly mutated in snakes. And this snake phenotype was put out in mice, and the mice came like limbless when they did that to the mice genome. So there is a relationship between losing this enhancer and limblessness. And it shows like a convergent trait between limbless amphibians and snakes. Either if it's the cause of the limblessness, because in some other lizards there are more complex ways of losing the limbs, so they have the enhancer intact there, but they lose the limbs as well. So it might be a conversion trait that is because the species are limbless, then this enhancer is free to mutate, and then they both lose it. But so if it's a cause or a consequence, we don't know, but we know that it's a shared conversion trait, that it's related to limblessness here. If you want to know more about the project and why we are sequencing everything and how we are approaching it, I also would like to point you to the PNAS series of papers that came out recently. The other thing I want to point out is that we do have this form that people can, if people are interested in specific species, specific taxa that occur in Britain and Ireland, and would then to be sequenced first, would like to prioritize them, people can ask us to. And if you have samples, is even better. Like because I mean, there is lots of laws on how like importing or exporting samples. No, so if you have like a bunch of species with lots of like good quality data and samples for species that occur in Britain and Ireland that we can import, that would be brilliant. That would be great. Better than like one species at a time, because we need to do all of the paperwork as well. But please suggest species that you are interested in. I also want to mention that a little bit on service. So I am a co-chair of the Justice, Equity, Diversity and Inclusion Committee of the Earth Biogenome Project, together with Anne McCarthy and Sadie Pius. So basically we are there to be a check and balance, right? Because as wonderful as it sounds, in terms of like we sequencing everything through this consortia, they can also be very centralizing in nature, right? And if you really want to sequence everything from lots of different parts of the world, we know the disparities that exist in the world. Lots of places that hold lots of biodiversity don't have infrastructure and funding enough to do the science. So how are we going to deal with that? And how are we going to make sure that we include everyone in a fair way in this project, right? So that's why we are there, and we work with wonderful people to always think through the questions of JEDI on biodiversity genomics. We have a paper coming out really soon. We'd really like your opinions on it when it comes out. I also want to invite you for Biodiversity Genomics 2024. This is an online, open, free-to-all conference. It's going to happen at the end of October. So it started being 100% online because of COVID, and then we decided to leave it because of inclusion, right? Because again, there is lots of people that cannot travel. So it's a very nice opportunity for PhD students and students that, you know, are unable to travel to present their work, and lots of people attend. It's very well attended by experts, so it's really nice, and I would like to invite you to join and have a look at the program coming up. And also I want to point out that we have, apart from Biodiversity Genomics, we have the Biodiversity Genomics Academy. We had one edition last year, which is basically also bringing lots of experts that have develop lots of different software to present on their tools, to explain people how to use it, to explain them what are the pitfalls, what are the, you know, like bugs or the weaknesses and the strengths of their softwares. So there is lots on genomics there already, and we are probably going to have a second edition of the academy as well, but there is a lot of material already available. So if you know a PhD student that wants to learn curation, genome assembly, and wants to learn the basics of it, I would like to you should point them to this Biodiversity Genomics Academy. Yeah, so with that, I conclude. So basically, we have been producing outstanding genomic data for the study of biology and evolution. So in this process, we have been also developing lots of different databases to make sense of all of this biological information, like Blob Toolkit and GOAT. They are all out there for the use of all. In the process, we've been also developing wet lab methods and bioinformatic methods, because it's a huge challenge. You need to automate and do things the best as we can. It's free information for the community to use. And apart from all of the evolutionary biology coming out of it, is also data for conservation in the time of the Anthropocene that we live in. So I want to thank you all for listening. And Mark Blackstad always said that it takes a village, and it's really like a village. It's a lot of people involved in this project. And I want to thank you for listening, and all of my colleagues at the Darwin Tree of Life project, especially Mark and Shane, which has been being my boss for the last 4 years. And thank you so much. And I'm open for questions.